So, you're sitting there listening to this, and, you probably feel pretty static, like your body is just a finished product.
Right. I mean, we all kind of feel like that on a day-to-day basis.
Yeah. But right now, literally, in the second it took me to say that sentence, your body just replaced roughly 2 million red blood cells.
2 million, just like that.
Just like that. And you're constantly healing microscopic tears in your muscles, too. I mean, you somehow grew from a single fertilized cell into an adult made of what? An estimated 37 trillion cells.
Give or take a few billion, yeah.
Right. So this relentless, completely invisible construction is happening constantly. And the engine behind all of this is a biological process. You probably remember the name of it from high school biology, which is mitosis.
Yeah, we really do tend to think of our physical stability as this default state. But that stability is actually a highly engineered illusion.
An illusion.
Yeah, because it's maintained by constant, meticulously choreographed cellular replacement. If that replacement stops, or, if it makes a mistake, the whole system breaks down really quickly.
Okay, let's unpack this because our mission for this deep dive is to figure out the actual mechanics of how cells pull this off. We want to go way beyond those basic textbook diagrams.
Definitely. Those diagrams leave a lot out.
They really do. So we're exploring the biomechanics of how a cell physically rips a copy of its DNA in half without destroying it, how this was even discovered, and the chemical fail-safes that keep us from turning into chaotic masses of rogue tissue.
Which is a very real risk, by the way.
Yeah, exactly. But before we get into the heavy machinery, I feel like we need to set a strict boundary for the listener regarding what mitosis is and, well, what it isn't, because people constantly confuse mitosis with meiosis.
Oh, all the time. It is easily the most common pitfall when discussing cellular biology.
Then the evolutionary stakes between the two are just completely different, right?
They couldn't be more different. So meiosis and mitosis serve entirely opposite genetic mandates. Meiosis is the division process that produces gametes of sperm in egg cells.
Right, the reproductive ones.
Exactly. Its entire purpose is to have the chromosome count and shuffle the genetic deck. It thrives on creating variety. But if we connect this to the bigger picture, mitosis is a strictly conservative division.
Conservative meaning it doesn't want any changes at all.
None. Zero. It produces 2 diploid daughter cells, meaning they retain the full, original paired set of chromosomes. The goal of mitosis is absolute genetic fidelity.
So it's preserving the exact blueprint of the parent cell.
Exactly, which is really the only way a multicellular organism can grow or repair specialized tissue without accidentally altering its own functional code.
That makes a lot of sense. So it makes the distinction purely functional. Like, if the tissue on my arm is damaged, my body needs an identical patch of skin cells. Any genetic creativity in that moment is a massive liability.
Precisely, because if a cell sorting error, what we call a non-disjunction, happens during meiosis, it can result in an entirely new organism carrying a systemic genetic variation, like trisomy 21 or Down syndrome.
Right.
But if a sorting error happens during mitosis, it usually just results in a localized cluster of dysfunctional cells. And your body usually either destroys those, or in the worst case scenario, it develops into a tumor.
Wow. Okay, so to maintain that incredible level of exact genetic fidelity across trillions of divisions, the cell has to do an immense amount of preparation before it even considers splitting, right?
Oh, a massive amount.
And that preparation happens in a phase called interphase. But historically, early microscopists looked at cells in interphase and called it the resting phase.
Which is just, honestly, an incredibly deceptive term. Cell biologists actively avoid using it now.
Because it's not resting at all.
Not even a little bit. Calling it the resting phase describes the visual absence of division, but it entirely ignores the biochemical reality. Interphase actually accounts for roughly 90% of a dividing cell's lifespan. Yeah. And it is the most metabolically demanding period the cell will ever experience.
Okay, let me make sure I'm visualizing the mechanics here. We are always taught to picture DNA as those little tightly packed eggshells.
Classic textbook chromosomes, yeah.
Right. But if the cell is metabolically active in interphase and doing its daily job like secreting enzymes or building proteins, it can't really do that if the DNA is tightly packed into a dense X, right? The genetic code would be physically inaccessible.
That is the exact crucial mechanical limitation. During interphase, your DNA exists as chromatin. It's this loose, diffuse, unspooled mass.
Unspooled, huh?
Think of it like reading the open source code of a software program. The cellular machinery literally needs to run along the strands of DNA to read the genes and synthesize proteins.
So if it were packed into those dense X shapes, it would be impossible to read.
It'd be like trying to read a heavily encrypted zipped file. You just can't do it. The X shape is strictly a logistical configuration for transport.
Oh, wow.
Yeah, it only condenses into that thick, armored state when it is time to physically move the genome.
So interphase is this highly active state where the DNA is unspooled. The cell grows during what we call the G1 phase, accumulating resources. Then it hits S phase, the synthesis phase.
Right.
This must be an absolute logistical nightmare. The cell has to copy its entire genome, all 3 billion base pairs, while the DNA is still loosely floating around as a mass of chromatin.
It really is an astonishing feat of molecular engineering. During S phase, replication enzymes run along the unspooled chromatin, creating a perfect duplicate of every single strand.
They don't get tangled.
Well, importantly, those duplicated strands remain loosely tethered together. Then the cell moves into the G2 phase, where it essentially performs a massive quality control check on the newly copied DNA.
We've got to make sure there are no errors before division.
Exactly. And in G2, it also begins synthesizing the actual physical microtubules, the microscopic cables, that it will use to pull the cell apart later.
So it's less like a cell resting and more like a manufacturing plant completely retooling its assembly line while remaining fully operational.
That's a great way to put it.
But not all cells stay on this assembly line, do they? The sources highlight this distinct offshoot called the G0 phase.
Yes, the G0 phase. And this is totally critical for human physiology. Many highly specialized cells, once they fully mature, permanently exit the cell cycle. They step into G0.
So they just stop dividing forever.
Forever. They continue to function, they metabolize, they do their jobs perfectly well, but the replication machinery is permanently dismantled.
Which explains the permanent nature of certain injuries. Like, if I cut my arm, the skin cells are actively cycling. They run through interphase and divide.
Right, they patch the skin right up.
But mature neurons in the brain or cardiac muscle cells in the heart, they're locked in G0.
That is the harsh biological reality that makes neurological trauma and myocardial infarctions, heart attacks, so devastating.
Because they can't replace themselves.
Exactly. A heart attack destroys cardiac tissue, and because those surviving heart cells cannot re-enter the cell cycle to undergo mitosis, the body can only patch the hole with non-contractile scar tissue. The engine of mitosis is just permanently parked for those specific tissues.
Man, that is grim but fascinating. Okay, so for the cells that do remain in the cycle, once they finish that massive duplication in S phase and the quality control in G2, they have to physically separate 2 identical genomes that are currently floating around as a loose, unspooled mess.
Which is no small task.
Right. Sorting that without snapping the strands must require serious mechanical force.
It does. It requires an entirely temporary dynamic cellular skeleton, which is what we see when we look at the actual visual stages of mitosis.
And to understand how we figured out these mechanics, we have to look back a bit, right? To 1882.
Yes, 1882. A German anatomist named Walther Flemming was trying to observe this process, but he had a major problem. Living cells are mostly transparent.
So you just can't see the gears turning?
Right, so he utilized aniline dyes, which were basically industrial colorants from the booming textile industry at the time, to stain the material inside the nucleus.
Oh, that's clever.
Yeah, and he actually coined the term chromatin, derived from the Greek word for color, simply because it was the stuff that absorbed the dye.
But he still needed to actually watch the process happen, right? The human cells divide way too fast to capture the mechanics with 19th century equipment.
They do. So Fleming brilliantly chose to study cells from salamander gills and fins.
Salamanders.
Yeah, because salamander cells are unusually large and they divide very slowly. So by applying these new dyes, he was able to sit at his microscope and meticulously sketch the mechanical progression of cell division over hours.
Wow.
He observed these thick, stained threads pulling apart and named the process mitosis, from the Greek word mitos, meaning thread.
Okay, here's where it gets really interesting, because the textbook version of mitosis prophase, metaphase, anaphase, telophase makes it seem like Fleming just looked through a lens and wrote down a completed manual in one afternoon. Oh, not at all.
Right. It was an incredibly fragmented international group project. Fleming proved the threads split lengthwise rather than just dissolving, but the actual stage names we use today were coined by a botanist, Edward Strasburger.
Right, because Strasburger realized the exact same mechanical process was happening in plant cells.
And then, a decade later, another anatomist, Martin Heidenhain, named the final stage telophase.
perfectly illustrates how scientific consensus accretes over decades. But by combining all their observations, we finally understood the incredible biomechanics of the early stages.
So in prophase, that loose chromatin finally tightly coils and condenses into the armored, transportable X shapes.
Exactly. And at the same time, two biological anchors, called centrosomes, migrate to opposite poles of the cell. They begin projecting a vast network of microtubules, essentially rigid protein cables, toward the center of the cell.
Forming the mitotic spindle.
Right. Then the cell enters metaphase. In human cells, this is where the nuclear envelope, the protective vault holding the DNA, completely dissolves. So the vault opens, and those microtubule cables invade the central space. But they aren't just blindly grabbing at the chromosomes, are they?
No, it's highly targeted. They attach to a very specific, highly complex protein anchor situated at the center of every single chromosome.
The kinetochore.
The kinetochore, yes. Think of it as a highly sophisticated biological winch. Each X-shaped chromosome consists of two identical sister chromatids, and they have kinetochores facing outward in opposite directions.
So the microtubules from the left pole of the cell attach to the left-facing kinetochore, and the cables from the right pole attach to the right-facing kinetochore.
Exactly.
Yeah.
Which beautifully sets up metaphase. The cables reel in the slack, dragging all 46 human chromosomes to the exact equator of the cell.
But this isn't just a passive alignment, like they're just hanging out in the middle.
Oh no, this is an intense microscopic tug-of-war. Both poles are pulling simultaneously, generating massive mechanical tension across the center of every single chromosome.
And that tension is the defining feature of metaphase, right? The chromosomes are suspended in perfect equilibrium.
Yes. It is visually the cleanest stage, which is actually why pathologists focus on metaphase when they need to count or analyze chromosomes.
Now, before we move to the climax of the division, the sources point out something wild. Our human method, dissolving the nuclear envelope so these cables can get in, is not a universal rule for biological life.
It is a profound reminder that what we consider standard biology is often just one evolutionary strategy. Humans and other animals utilize what's called open mitosis, breaking down the nucleus.
Right.
But many fungi, including yeast, utilize closed mitosis. Their nuclear envelope never dissolves.
Wait, so how do the cables get in?
They don't. The centrosomes and the entire spindle apparatus form inside the intact nucleus. The chromosomes segregate within a completely closed vault, and then the nucleus itself just pinches in half.
That's crazy. It's a completely different engineering solution to the exact same problem.
Exactly. But regardless of the vault being open or closed, the physical separation has to happen. That is anaphase. The snap. The proteins binding the two halves of the chromosome snap. The tension from the spindle cables violently rips the identical sister chromatids apart, pulling one complete set to the left pole and one complete set to the right pole.
Which is followed immediately by telophase. The cell now has two identical genomes at opposite ends. A new nuclear envelope begins to construct itself around each set, and the tightly coiled chromosomes unspool back into diffuse chromatin so the genes can be read again.
Right. And we really need to clarify here that mitosis itself is now technically over.
Wait, really? But the cell hasn't split yet?
I know, but mitosis is strictly the division of the nucleus, the genetic material. The actual physical splitting of the cell body into two distinct cells is a completely separate mechanical process called cytokinesis.
Oh, okay.
Yes, cytokinesis overlaps with the end of mitosis, but it utilizes totally different machinery. In animal cells, a contractile ring of actin and myosin proteins acts like a microscopic drawstring, literally physically pinching the cell membrane in half.
But plant cells can't do that, right? Because of the cell wall.
Right, because they have a rigid outer cell wall. They can't pinch. They have to essentially build a new brick wall called a cell plate straight down the middle to divide the space.
That's fascinating. But going back to the cables and the kinetochores, if these microtubules are just casting out like fishing lines to catch these anchors, the error rate should be catastrophic.
It intuitively seems like it would be, yeah.
I mean, if a cable misses its mark or attaches poorly and the cell divides anyway, you're going to rip a chromosome into pieces or pull an entire pair to one side. There has to be a master control system.
There is. And for a century after Fleming, it was a complete mystery. We could see the gears moving, but we couldn't find the engine.
So what changed?
That changed late in the 20th century with the discovery of the cell's biochemical pacemaker. Cyclins and cyclin-dependent kinases, or CDKs.
CDKs.
Right. These are regulatory proteins whose concentrations rigidly rise and fall, creating a chemical clock that dictates exactly when a cell is allowed to transition from one phase to the next.
And baked into that clock are the fail-safes. The most critical one during division being the spindle assembly checkpoint, the SAC.
The SAC operates precisely during that metaphase tug-of-war we talked about. You can think of it like a highly sensitive tension sensor on a seatbelt.
Okay.
The SAC physically halts the entire division process. It prevents the onset of anaphase until it chemically verifies that every single kinetochore is attached to a microtubule, and more importantly, that there is equal opposing tension on every single chromosome.
So it does the math on the tension.
It does. Only when the tension is mathematically perfect does the SAC release the brake.
Because if the SAC fails, you get aneuploidy. A cell divides while missing a cable connection, resulting in one daughter cell receiving extra chromosomes while the other is short-chained.
Exactly. The genetic blueprint is totally corrupted. And that systemic corruption, aneuploidy, is a primary hallmark of almost all solid cancer tumors. Yeah. When we discuss cancer, we are fundamentally discussing a disease of mitotic regulation. The genetic mutations in a cancer cell specifically disable the cyclins, the CDKs, and the SAC. The biological brakes are just cut, and the cell undergoes rapid, relentless, unchecked mitosis.
Which is terrifying, but that actually raises a really important point. If we have mapped this pacemaker, if we know exactly how the SAC and the cyclins operate, why is chemotherapy still such a systemic, destructive treatment?
This raises an important question because it really comes down to the mechanics of the drugs.
I mean, we use drugs like paclitaxel or vincristine, which incredibly are derived from the Pacific yew tree and the Madagascar periwinkle, but they seem to act like a sledgehammer, causing such intense side effects.
They do, and it highlights a massive limitation in pharmacological targeting. Those specific chemotherapy drugs are known as microtubule poisons.
Microtubule poisons, so they target the cables.
Yes. They physically bind to the tubulin proteins that make up the spindle fibers. Depending on the drug, they either freeze the cables in place so they can't reel in, or they prevent the cables from forming at all.
So the cancer cell is trapped.
Exactly. It's indefinitely trapped in metaphase by its own SAC, and eventually it just triggers its own death sequence. The problem is these drugs are entirely blind.
Right. They can't distinguish between a rogue cancer cell and a perfectly healthy cell. They only target the mechanism of mitosis itself.
Exactly. And your body relies on rapid mitosis to survive. The epithelial tissue lining your gastrointestinal tract is a prime example.
Because digestion is so abrasive.
Highly abrasive. So to maintain integrity, stem cells in your gut undergo relentless mitosis, completely replacing your entire intestinal lining every 4 to 5 days.
Every 4 to 5 days? That's insane.
It's a lot of division. So when a patient receives a microtubule poison to halt a tumor, that drug simultaneously freezes the mitotic spindles in their gut stem cells.
Oh, no.
Yeah. The intestinal lining cannot renew, the tissue degrades, and the patient experiences severe nausea and digestive toxicity. It is a stark demonstration of how deeply we rely on continuous cell division.
Understanding those fail-safes and figuring out how to chemically freeze the spindle is exactly what finally gave researchers the leverage to manipulate the system and correct our own biology textbooks, though.
Oh, absolutely. It literally allowed us to properly count our own genes.
This part of the source material blew my mind. For 30 years, from the 1920s through the early 1950s, the scientific consensus was that human beings had 48 chromosomes.
48, yeah. That was the accepted fact.
Which is staggering. The entire scientific establishment was wrong about a fundamental integer of human biology. They were looking at complex, tangled spreads of chromatin under rudimentary microscopes and simply miscounting.
They were just doing the best with what they had. But what's fascinating here is how their correction was ultimately a triumph of chemical manipulation, not just better optics. In 1956, researchers Joe Hingio and Albert Slavon utilized a compound called colchicine. Colchicine acts much like the chemotherapy drugs we just discussed. It actively depolymerizes microtubules, basically obliterating the mitotic spindle.
So by applying colchicine to human lung cells, Hingio and Levin chemically arrested thousands of cells at the exact moment of metaphase.
Precisely. They trapped the chromosomes right after they condensed into their thickest, most visible X shapes, but before they could be dragged together into a tangled mess at the equator.
So they were just floating there, perfectly frozen.
Frozen and swollen. The chromosomes were maximally condensed and spatially separated. And with these pristine, arrested visual spreads, they could easily and definitively count exactly 46 chromosomes, permanently correcting the record.
It just proves that in cellular biology, your understanding is inherently limited by your ability to freeze the mechanics.
Very true.
Which brings us to the ultimate example of manipulating those mechanics, the HeLa cell line. In 1951, a sample of aggressive cervical cancer cells was taken from Henrietta Lacks. And normally, human cells have a strict, built-in biological countdown timer known as the Hayflick limit.
Right, the Hayflick limit. It's actually a really elegant defense mechanism.
Right.
Every time a cell copies its DNA during S phase, the replication enzymes physically cannot copy the very last few base pairs at the end of the strand.
Like the copier runs out of track.
Exactly. It's a mechanical limitation of the enzyme itself. So to protect the vital genes, our chromosomes have long, repetitive junk sequences at their tips called telomeres.
Like the plastic caps on shoelaces.
Perfect analogy. But with every round of mitosis, those telomeres get shaved down. After about 40 to 60 divisions, the telomeres are critically short. The cell senses this, halts the cell cycle, and promptly retires.
Which prevents old, potentially mutated cells from continuing to replicate.
Exactly.
But the HeLa cells had a specific mutation that reactivated an enzyme called telomerase.
And telomerase is a remarkable enzyme. It carries its own RNA template, it actively binds to the ends of the chromosomes, and chemically synthesizes new telomere sequences, rebuilding the protective caps as fast as they are shaved off.
So in Henrietta Lacks cancer cells, this enzyme was just hyperactive.
Extremely hyperactive. The countdown timer was permanently refilled. Those cells completely bypassed the Hayflick limit entirely.
Meaning the HeLa line has been undergoing relentless mitosis in laboratories globally for over 7 decades.
Unstoppable division.
They were instrumental in testing the polio vaccine, mapping the human genome. I mean, they are the ultimate biological proof of concept for immortal cell division.
They really are. But they also demonstrate the terrifying, boundless power of the mitotic engine when it is decoupled from the regulatory checkpoints.
Definitely a double-edged sword. All right, let me synthesize the core mechanics we've explored today. First, mitosis is about genetic conservation. It uses a complex spindle apparatus to perfectly duplicate and divide the genome for growth and repair, unlike meiosis, which shuffles the deck. Second, the famous X-shaped chromosome is strictly a tightly packed transport configuration used during division. Normally, your DNA is unspooled chromatin, so the cell can actually function.
Yes.
Third, this entire physical dance is rigidly controlled by a master chemical clock of cyclins and CDKs and heavily monitored by tension-sensing checkpoints like the SAC. And finally, when those checkpoints fail or telomeres are artificially rebuilt, you get the unchecked division we recognize as cancer.
That is an excellent mechanical summary. And to close, I actually want to pose a theoretical scenario based on what we've unpacked about the cell cycle and that G0 phase we talked about earlier.
Okay, I'm listening.
We discuss how human cardiac cells and central nervous system neurons are permanently locked out of the mitotic cycle, making heart attacks and spinal cord injuries permanent.
Right, because the replication machinery is dismantled.
Yes. However, the human liver is a major exception. It is one of the few organs capable of massive, rapid regeneration.
Oh, really?
Yeah. If a patient loses a significant portion of their liver, the surviving hepatocytes, the liver cells, can instantly re-enter the cell cycle, triggering a massive wave of localized mitosis to rebuild the lost tissue mass.
Wow, so it just turns the engine back on?
Exactly. So the profound question for the future of regenerative medicine is this. As our understanding of cyclins, CDKs, and microtubule regulation becomes absolute, is it mechanically possible to selectively unpark the engine?
Oh.
Right. Could we one day safely, temporarily reactivate the mitotic cycle in a damaged spinal cord or a scarred heart, allow the tissue to regenerate perfectly, and then chemically re-engage the brakes before it spirals into cancer?
That implies a future where we don't just treat the symptoms of permanent tissue damage, but we actually learn to pilot the master clock of the cells themselves to heal the currently unhealable. That is a massive paradigm shift.
It truly is. It's the ultimate frontier of cellular engineering.
Well, thank you so much for joining us on this deep dive. The next time you find yourself sitting still feeling like you haven't accomplished much, take a moment to realize that underneath your skin, you are a relentless, highly regulated biological construction site. You've got billions of cells verifying mechanical tension and pulling cables just to keep you intact. Take care, everyone.